Our main topic for the day is blood grouping or blood groups and the resource factor. Now, we are all going to know what blood groups are and the importance or why we all have to know our blood group. Then after that, we're going to do an experiment, and this experiment we call it cross matching, why you can't give a strange blood or you can't give blood to anybody just like that, why it is very important for us to do cross matching. Please, let's tap on the screen. If you are just joining, follow me if you've not followed me. Okay, so when we are talking about blood grouping, or when we talk about blood grouping, we are mostly talking about the ABO blood grouping. And this ABO blood grouping was discovered by a scientist in the year 1901. And the name of that scientist is Carl Landsteiner. Okay, he was an Australian scientist who discovered the ABO blood grouping. Now, we have different other blood grouping or blood types that are there, okay, but the one that is more immunogenic, the one that we react to, is the ABO blood grouping. When we talk about blood grouping, we can talk about the Delphi, the Lewis, the Bombay, MNS. There are so many blood types we can talk about, or blood grouping systems we can talk about, but the one that is of interest to we human beings are the ABO blood grouping. The ABO blood grouping. Now, the person who discovered the blood grouping in the year 1901 classified this blood grouping in 4 sections, okay? So he classified the blood groups into blood group A, blood group B, blood group AB, and blood group O. So these are the various blood groups or blood types you are coming to talk about. Okay. Now, I can see a lot of people are living their life because they don't see the importance of this. I intentionally brought this sample to show it under the microscope, and a lot of people were joining the live because we are coming to see something under the microscope. But I'll tell you, if you are here, tap on your screen because this is something we must all know. Okay, now let's draw a table, and please, this table is not for fun. This table is going to help us understand blood grouping and also the practicals we'll be doing. Okay, this table is going to help us understand the blood grouping and the practicals that will be involved. Please, let's all tap on the screen if you want to follow what we are doing. Okay, all right, so far only 2 people share the live because you don't see the importance of this. That is why we see only 2 people sharing the live. All right, so we have blood types here. So blood groups, this is our blood type or our blood groups. Okay, now in blood grouping, there are some certain terms we use. We use a term like antigen and antibody. Antigen and antibody. These are the various tests or terms we will be using. Okay. Now, the person who discovered the blood grouping defined this antigen and antibodies for the blood grouping. Now, he said antigens for blood groupings are glycoproteins. They are sugars and they are proteins that are found on our red cell surface. Okay, he said antigens for our blood groupings are sugars and also proteins that are found on our red cell surface. Now, those who haven't seen the blood before, this is our blood. Okay, this is the blood, and what you see here is a blood that has separated. Okay, a blood that has been separated. Now, our blood contains 4 main components. There are 4 main components in our blood. and I know you guys, we all did science in JSS, that is basic school, in junior high school, in senior high school. So if I say the component of the blood, we all know the basic component of the blood. Who can mention the basic component of the blood? There are about 4. There are 4 basic components, okay, of the blood. Let's mention them here. Please, let's tap on the screen. Follow me if you've not followed me, okay? And let's learn together. So big D is saying platelet. That is 1. Beautiful. Big D is saying platelet. Okay. Nikilav is saying plasma. That is beautiful. So now we have 2. Momita, you're welcome. Red blood cell. Now we have 3. Somebody is mentioning serum, the only legend. All right. So serum stroke plasma. We'll come and talk about why serum or plasma. So leukocytes, very good. So leukocyte is a big term. So leukocytes are the same as what? White blood cells. Okay. So when we are talking about the basic component of the blood, we are talking about the plasma or serum. We are talking about the red blood cell, which is the RBC. We are talking about the white blood cell, which is the WBC, and the platelet. Now, this is our blood, and all the components of the blood are present here. When you check the top here, what you see at the top here, or the yellow thing you see at the top here, is the liquid portion of our blood. Our blood is not red. Our blood is not red. It is our red cell that is red. The blood itself is not red. It is the red cell that is red. And even the red cell itself is not red. It is what found in the red cell that makes the red cell red. And who knows that thing in the red cell that makes the red cell red? Who knows the thing in the red cell that makes the red cell red, and that thing makes our blood looks red also? Hemoglobin. Okay. Hemoglobin. Now we'll come and talk about that. Hemoglobin is what carries oxygen to all parts of the body. So hemoglobin is a protein which is very, very, very important in our blood. Now when you check this, the liquid portion of the blood is the straw liquid thing you see here. Okay, that is the liquid portion of our blood. Now at the down here is our red blood cell. The down here, and you can see it's red. That is our red blood cell. And in the middle of the liquid portion of our blood, okay, in the middle of the liquid portion and also the red blood cell are the Buffy coat. And when you check this and when you pay attention, you see that there is a white coat, something lining in between the plasma and the red cell. Please, can we see that? Can we see that in between the red cell and the plasma, there is some whitish substances? Okay, we call it the Buffy coat in between there. Yes, that is where the plasma and the red cells are located. So the red cells and the the white cells and the platelets, the white cells and the platelets are in the middle here. This is our red cell and this is our white blood, our plasma. Okay, our red cell, our plasma, the platelet and the white cells are in the middle here. Now, doing blood grouping, we said antigens are glycoproteins. Mandela, God bless you. Antigens are glycoproteins that are found on our red blood cells. Okay, antigen for the blood groupings, they are proteins and also sugars, and they are found on the surface of our red blood cell. So let me show you guys the red blood cell. Maybe those who haven't seen the red blood cell before, let's watch the red blood cell under the microscope. Let's watch the red blood cell under the microscope. So let's make a small smear. I know it's going to be some way, but let's try and see. So coming to view our red blood cell under the microscope. When was circulating fluid when? Okay. Since I can't see my circulating fluid, let me use normal. Please do what to follow me if you've not followed me, okay? Do what to follow me if you've not followed me. Let's tap on the screen. We are here to learn, okay? On my platform, we learn every day. Oh, fuck yeah. So we're coming to view our red blood cell under the microscope. Those who haven't seen the red cell before, let me show you how the red cell looks like and where the antigens are found. Good. So there are millions of red cells here. And these red cells are created. These red cells are created. The round things you see, those are what? Created red cells. Those are created red cells. Okay, those are created red cells. So those are the red cells. They are cells, they are living organisms, and you can see them move. I should wear gloves, okay. So those are the red cells. And we are saying that our antigens for the blood grouping, Sarah, you're welcome, my dear. How are you? Are located on the red cell surface. Okay, today we have so many practicals we'll be doing, so let's be fast. We'll be closing early today. Okay, now he defined antibodies for blood grouping as immunoglobulins that fight against foreign antigen. So antigens are glycoproteins on our red cell surface, and antibodies are immunoglobulins, or they are armies, they are soldiers that are on our inside our blood, and they fight against foreign blood. Okay, so that is the definition for antibody for our blood groups. Now, before we can fill this table, we need to study a principle made by what? The person who discovered the blood grouping, Karl Landsteiner. He made a principle in the year 1901, and this principle says that all blood groups or types produces antigen of self, but Naya, who's that? Naya, what did I do to deserve that? What did I do, Naya? Naya, God bless you. Please thank Naya for me. I've not seen anything like this before. Please, everybody, thank Naya for me. God bless you, Sister Naya. My God. I don't even know what to say. How? What is happening? Naya, God bless you, Naya. God bless you. Guys, I don't even know what to say. Naya, I'm grateful. God bless you. I've not seen anything like this. See, I've been doing live for 8 months now. I've not seen anything like this before. I've not seen anything like this before. God bless you. Oh my goodness. I thank you so much. God bless you. Thank you so much. Guys, I'm grateful. Wow. Okay, let's continue. Naya, I don't know how much I would thank you. Guys, Naya don't want me to continue the live today. Naya don't want me to continue my live today. Naya don't want me to continue my live today, guys. I'm going home, guys.